was a great opportunity to hear him give exceptional talk. His first talk on the auditory system, and now he's going to talk to us about myelin and oligodendrocyte. And Dr. Dwight is a professor of neuroscience and otolaryngology, head and neck surgery at the Johns Hopkins University School of Medicine. And his research has focused on synaptic physiology, glutamate transporters, and glue involvement in neuronal signaling. And he has done his PhD in Stanford University.
Love today, which was studies of oligodendrocytes and their progenitors, which kind of happened serendipitously when I was a graduate student, and I started to record from glial cells whose identity I didn't know at the time I hit the campus. And these turned out to be oligodendrocyte progenitor cells, but amazingly enough, it turned out that these cells form direct synapses with neurons, and that sort of started my interest in oligodendrocytes and myelin biology. So just to make sure that everyone is on the same page, start this page there. Right, so oligodendrocytes are obviously the myelin-forming cells in the central nervous system. They are abundant in both gray and white matter. They extend these fine cytoplasmic processes, which then extend these lamellar sheets that wrap around axons, and each oligodendrocyte makes maybe 30 or more of these discrete segments of myelin along axons. And so these are cells that we all know are important for accelerating the propagation of action potentials along axons by reducing capacitance and also increasing the membrane resistance. But over the last few years, it's become clear that myelin and oligodendrocytes are not just these kind of inert insulation in the central nervous system, that they are actively involved in regulating excitability of cells and providing metabolic support. In fact, these studies have shown that they also regulate acid-permeated stasis and can control excitability of neurons. And we showed 3 years ago now that if you delete a specific type of ion channel, an inwardly rectifying potassium channel known as KIR 4.1, exclusively from the oligodendrocytes, these animals present with uncontrolled seizures and eventually will die prematurely. So clearly, these are active cells that play an important role in regulating not just the conduction of action potentials, but also circuit function by controlling ion homeostasis and metabolic support for axons. Now, our knowledge of oligodendrocytes comes primarily from studies that have been done in white matter. White matter looks white because it has a lot of myelin and reflects light. But as you can see in this section of a mouse brain, an adult mouse brain that has been immunostained for myelin basic protein, all of these white areas highlight the regions of myelin. And clearly, the cortical mantle is not devoid of myelin, although it's referred to as gray matter. In fact, there's lots and lots of oligodendrocytes which are present here, which form myelin and wrap around various types of axons. Now, what you can also appreciate from this section is that myelin in the gray matter is not uniform. And our studies of myelin in the gray matter are kind of challenging long-held beliefs about the general organization of myelin and the properties of oligodendrocytes. And so one of the things that's quite dramatic about myelin in the gray matter is that, as you can see here, it's not uniform. So there are regions which are have tens of large numbers of oligodendrocytes and myelin, and then there are these regions which are essentially largely devoid of myelin, but very few oligodendrocytes. That's quite difficult in the association areas versus the sensory areas which have higher degrees of myelination. Now, let's add a little bit more to the complexity here. And it turns out that in gray matter, the pattern of myelin along axons that are formed from particular cell types is also variable. So you might imagine that all this variability could be due to different subtypes of cells. But what we can see when we do the histology is that even for a given cell type, so here we've genetically labeled a subset of thalamocortical neurons, and those are shown in red. And in cyan are the myelin sheaths. And you can appreciate in this adult tissue that even axons that are located right next to each other, some can have myelin and some will be devoid of myelin. So it's highly variable among the axons of discrete cell types. And to take this even one step further, it turns out that myelin in the cortex is often discontinuous. That is, that there are segments of myelin that are often separated by these large gaps where there's no myelin. So myelin is highly variable in the cortex. Individual axons of discrete cell types can exhibit different patterns of myelin, and even along individual axons, the pattern of myelin can be discontinuous. So it's a very striking, highly variable pattern of myelin that's formed in brain matter, which is raising lots of questions about how this pattern is generated and what happens to this pattern over time. But you can appreciate right away, because not everything is myelinated, it presents basically a substrate that could be used to generate a lot of plasticity, if you will, in the nervous system. So that's generating a lot of excitement in the field that the modification, formation of myelin could be a new form of plasticity that's important for things like motor learning. Now, how do we study myelin? Obviously, we can use histology, like I just showed you. These are sections of human brain, actually, from control patients and patients that died because they had multiple sclerosis. You can see that there's a large region of the cortex here, which is devoid of myelin. And at a higher resolution, we can use stains like myelin-basic protein or proteolytic protein in this case to visualize the myelinating cells from the pre-myelinating stage to the true mature oligodendrocyte. But of course, these are only in ex vivo, tissues that have been taken out. They don't really tell us much about the dynamics of the cells and things like that. So the primary means that people have used to study the dynamics of myelin has been through techniques like magnetic resonance imaging. This is a movie longitudinal imaging of an individual where you can see that there are these foci of demyelination that resolve over time. And so this gives us an indication of the disappearance or loss of myelin and then regeneration of myelin in the nervous system. So extremely powerful approach for looking at myelin dynamics in this clinical setting. But of course, one of the challenges with this approach is that it has very, very low resolution. So one voxel, so the individual unit of resolution in the MRI, has about 700,000 oligodendrocytes. So this doesn't really tell us anything about the dynamics of the cells individually. This is the rich data that we need to understand to learn how these cells regenerate themselves and how they modify the nervous system over time. So the approach that we've used is really to bring this into the realm of cellular resolution, and to do this we use a technique known as photon imaging, which I'm sure you're all aware of. It's a microscopy technique which allows us to visualize cells within scattering media, and this approach is applied here in combination with transgenic mice. In this case, it's a mouse that expresses enhanced progressive protein under control of the MOBP, or myelin oligodendrocyte basic protein. It's a component of myelin. It's not MBP, but it's actually a very abundant protein in myelin. And this mouse line was generated by the GenSat project. It's a bacterial artificial chromosome. The only thing that's relevant here is that this allows cytosolic GFP to expressify each and every oligodendrocytes in the brain. And then we make cranial windows on these mice. We remove a portion of the skull, replace it with a cover slit, and glue that into place. And then the animals will, live their full lifespan. But we have a window now on the brain, and we place those animals underneath one of these microscopes, and it allows us to visualize the oligodendrocytes. Now, one of the things about oligodendrocytes in the cortex is that nature has, for once, been very kind to us in that there are a number of fibers that are oriented tangential to the surface of the cortex, And as a result of that, the oligodendrocytes also adopt this morphology. So they're almost planar, where they extend their processes in a plane and they form myelin sheaths. And that allows us better resolution of those processes, because the XY resolution is better than the Z resolution using this type of microscopy. And so if you look from the top-down view, you can see the individual oligodendrocytes and their myelin sheaths in these mice. And if you look at the Z projection after taking a stab, you can appreciate that here up near the cortical surface, there are a number of these planar oligodendrocytes, and as you go deeper, the cells become vertically oriented as they align to the ascending axons that are entering the cortex. So what does this look like in a living animal? I'll show you a movie here as we pan down from the cortical surface down into the cortex, and you can see the individual cell bodies of oligodendrocytes. And now as you go deeper into the cortex, you see these little points of light, because you're basically sectioning through the individual sheaths that are oriented vertically. And what is quite amazing about the resolution in this system is that you can visualize individual oligodendrocytes and essentially every single myelin sheath that they form. So it's such a massive contrast to things like MRI. It's basically giving you histology in living animals, where we can see this rich diversity of their morphology in different brain regions. So what exactly are we actually looking at here when we are looking at the oligodendrocytes? Well, here you can see that there's one cell, and it's extending a cytoplasmic sheath, which then encounters an axon, and then extends a myelin sheath along this axon. Here's another cytoplasmic sheath which extends a myelin sheath. And you can appreciate that this is not particularly uniform. Remember, we're using cytoplasmic GFP. So what we're actually looking at here are the inner and outer tongues of the myelin, where there is substantial cytoplasm. Because remember, myelin itself is compact membranes. There's no cytoplasm really in there at all. And so if you have cytosolic GFP, you would not see any fluorescence there, because basically it's extruded. So the regions that we see GFP is the regions where there are cytoplasm. And there are these little channels that go down the myelin, and in particular the perinodal regions, where you see the expansion of the cytoplasm here. So those are the areas where the GFP accumulates and then we can visualize. But importantly, it gives us an image of the full extent of the association of these cells with the axon. That is, it gives us an indication of the distribution and length of the individual myelin sheaths that are formed by these cells. This is an example of the inner and outer tongue. Okay, so the first question we had is, what are the dynamics of oligodendrocytes in the cortex? And so we imaged animals longitudinally, and this approach allows us to take an image of the cortex, we take a volume, and then we put the animal back in the animal facility, and then we'll bring them back at various intervals to then visualize what's happening. And we can go back to the exact same place, and we can see the exact same oligodendrocytes and even the exact same myelin sheaths over many months. So it really gives us the ability to then start to study things like the dynamics of the cells. So in an animal that was 2 months of age, you can see here's the interval of imaging: 1 day, 2 day, 3 day, 4 day. And on the 4th day, or the interval between 3rd and 4th, there was the appearance of a new cell. So in an animal that was 2 months of age, we can see the generation of new oligodendrocytes. That's what you would expect, because oligodendrogenesis is thought to have a pretty extended developmental time course. And what about in an animal that's 8 months of age? So here's 0 days, 30 days, and in that interval we saw the appearance of a new cell, 44, 46 days, and indeed there's another cell that appeared in this interval. And all of these 3 cells were persistent in this volume throughout the imaging period. So even in an animal that was 8 months of age, we can see the formation and integration of new oligodendrocytes within the cortex. And it turns out that if you sort of project this over time, and you can do this histologically as well, one thing that's really fascinating about myelination and oligodendrogenesis is that it does have this extremely extended time course. So here you can compare the distribution and density of oligodendrocytes in an animal that's 3 months of age with an animal that's 10 months older or 21 months of age. So you can see that between 3 months and 13 months, there's a massive increase in the number of oligodendrocytes. And we can look at this quantitatively here and also in relation to different layers of the cortex. And so between three months to a year, there's basically a doubling of the oligodendrocytes that are present within the cortex. And this is pretty surprising to most people because most people consider like a 3 to 4 month old mouse to be adult. I mean, it's sexually mature, and many behavioral studies are done on mice that are that age. But what you can see here is that an animal that's that age has only half of the oligodendrocytes and presumably half or less of the total myelin that it will have. So it's quite immature when it comes to myelin. So I think that's a sort of a fascinating developmental change, that we have to take into account when we're doing behavioral studies. So the appearance of new oligodendrocytes in the cortex, I think, in some ways is very satisfying because it provides an explanation for the persistence of the progenitor cells over time. And what I'm showing you here is sort of hemisections of the brain from 2 different transgenic mice, one on the left, in which all of the oligodendrocyte progenitors are visible, and the one on the right where the oligodendrocytes are visible. And what you can appreciate on the left is that there's a huge number of these oligodendrocyte precursor cells that are maintained in the adult nervous system. So they make up about around 5% of all cells, and you can see that they have this really highly ordered and tiled distribution where cells are spaced about equidistant from each other. And this distribution is established early in life and is persistent throughout life. So it's an amazing situation where you have progenitor cells that are widely distributed in the CNS and they're maintained throughout life. So it's very unlike neural progenitors and things like that. And these cells we've shown are lineage restricted, in the sense that they either remain in this progenitor state or they mature and become oligodendrocytes. So the fact that new oligodendrocytes are being generated in these much older animals provides an explanation for why these progenitor cells are maintained throughout life, because they continue to generate new cells. Now, an alternative explanation for that might be that, well, maybe oligodendrocytes don't live forever. And if they don't, then you might imagine that you would have to have progenitor cells around to replace them because they have these critical functions in the nervous system. And if this is a random process where you don't know where and when an oligodendrocyte is going to die, then you'd have to have progenitor cells located all over the place. And so we then did a number of experiments looking at the stability of oligodendrocytes. And this is an experiment that was done over almost 2 months, and there were individual oligodendrocytes. This was done in a year-old mouse. You can see these cells here. And so we tracked those oligodendrocytes over time, and here's an overlay of the 0 days and 50 days, and the white cells are ones where there's exact overlap between the cells. You can see that there was one new oligodendrocyte that appeared in this interval, but all of the others are on top of each other. And so it's pretty remarkable. I think we've done many thousands of oligodendrocytes tracked in many animals. In this case, it was 5 different animals. This data set had almost 1,000 oligodendrocytes, and essentially every single cell that we imaged at time 0 was still there 2 months later. And it didn't matter really which age animal we were looking at. It revealed that oligodendrocytes, once they're generated and integrated, are extremely stable. And basically, the only thing that you see is in these animals the generation of new cells, where none of the oligodendrocytes, once they appear in these transgenic mice, once they reach that stage where the MOBP promoter is active, and GFP is expressed, and myelin is formed, they're very, very stable. So essentially, there's very little turnover of oligodendrocytes, and suggests then that the progenitors are used to generate new cells. So this is one form of plasticity. But of course, there could be lots of other forms of plasticity that involve reorganization of the myelin sheaths. You can have cases where the sheaths increase their length. You can have generation of new sheaths, lost sheaths, or shrinking of sheaths. I mean, there's many combinations of kinds of remodeling of sheaths that you might imagine. And so we used these transgenic mice that to look really with high magnification out at the individual sheaths. And it's remarkable in these animals, you can even see nodes of Grandier where the DFP accumulates in these perinodal regions. So there's a couple nodes of Grandier that are visible. And so we tracked these different sheaths. And so here's an example of two sheaths that are forming a node of Grandier in this year-old mouse. It's extremely stable over 48 days. And this stability even carried over to these isolated sheaths. Remember I told you that in some cases there's discontinuous myelination along axons, and you might have just an isolated myelin sheath with no sheaths flanking it. Even these isolated sheaths were extremely stable. Now we did see some reorganization of sheaths, so here's an example of an extension of a sheath and a retraction of a sheath. A number of animals, 6 mice in this case, we found about 6% of myelin internodes were remodeled. So maybe that's significant. We didn't really know at the time. But subsequent to this, what we realized is almost all of this remodeling was due to remodeling of newly generated cells. And so what we find is that once new oligodendrocytes are generated, there's a period of plasticity. And those new cells extend sheaths, and some of those sheaths can be trimmed away, some extend, some shrink, things like that. So there's a period of plasticity, and here's a plot showing the length change over time from the first appearance of the cells. And you can see over time there's a gradual decrease in overall length of myelin. As basically the cells are remodeling their myelin sheaths. And so the plasticity that we saw sort of at first blush in these initial experiments really had to do with the newly generated cells that were reorganizing their sheaths. But after this 2-week period, they become really like locked in into these circuits, and they don't really show this kind of like extension or retraction anymore. So what we conclude from this are that oligodendrocytes and myelin are formed throughout life in the cortex, and then after an initial period of remodeling, the myelin sheaths are extremely stable. So you might say, well, these are animals that are not in any kind of enriched environment, they're in a boring cage. Maybe if you challenge the animals or altered their environment, you would see some reorganization. So what we did is we basically sent the mice to Mardi Gras. We had developed cages that had beads on strings that were hanging down from the cage, and when the animals walk through the cage, it hits their whiskers. And so this has been shown to induce a lot of structural reorganization of dendritic spines in the somatosensory cortex. So it's a known mechanism to induce plasticity in the somatosensory system. And so we replicated this, and we asked, will this induce any plasticity in myelin? And again, we looked at the individual myelin sheaths and to see, basically here's examples of isolated sheaths. Sometimes they increased a little bit, sometimes they decreased. But overall, it was surprising that there was essentially no change in the number of retractions, extensions, or percent of the myelin internodes that were dynamic in these animals. So even though there's a dramatic change in their environment, we're stimulating a lot of activity in the somatosensory system, this did not induce any concerted change in the individual myelin sheaths. Now what about the generation of new oligodendrocytes? So of course we can track the individual cells, and in a control mouse, we will occasionally see new oligodendrocytes being generated. This was a 3-week interval. And here shows an example that was subjected to sensory enrichment. In this example, we saw 2 cells that were generated within this field of view. If we cut the whiskers from the animals, we don't see any loss in the oligodendrocytes once they're generated. So again, this reinforces the idea that these cells, once they're generated, are extremely stable. So sensory deprivation did not result in turnover of the oligodendrocytes. The only thing that changed the number of oligodendrocytes was the enrichment itself. So control mice have a certain number of oligodendrocytes that are generated. This was not altered by anesthesia. The animals that were in the enriched environment increased the production of oligodendrocytes. If we trimmed the whiskers and then put the animals in the enriched environment, we did not see a change in the number of oligodendrocytes that were generated. And as I mentioned, if we deprive the animals, there's no change. So it does appear that life experience can enhance the generation of new oligodendrocytes. So we conclude from this that changes in myelin occur primarily through the generation of new oligodendrocytes. It's not through loss of the cells or the existing cells extending new processes that then sheath new axons. It's really by the de novo production of new oligodendrocytes. So that's what's happening in a normal brain. What happens under regenerating conditions? And so I'm sure you're all aware of the disease multiple sclerosis. This is a disease in which the immune system attacks myelin and it induces the death of oligodendrocytes. And this disease typically presents in a remitting relapsing form initially, where there are bouts of demyelination that are then followed by recovery as new oligodendrocytes are regenerated. But unfortunately, this disease often transitions to a progressive phase in which there's no longer any regeneration of oligodendrocytes, and you have chronic lesions that exist and substantial increase in cognitive deficits, motor deficits, and things like that. And it turns out that demyelination is very common in the gray matter, although it's been studied a lot less because in MRI it's actually difficult to detect myelin in the gray matter because it's often highly disordered, and MRI is really looking at the nature of water diffusion. But histologically, you can see that there's a lot of demyelination that occurs in gray matter. In fact, 93% of MS brains contain cortical lesions, and it's highly correlated with the degree of cognitive decline, as you might imagine. So we used a model in which we can feed animals an oligotoxin known as cuprizone. This is a copper chelating agent which oligodendrocytes are very sensitive to and causes them to degenerate. And so the general protocol here is that we put the animals on cuprizone feed for a period of about 3 to 6 weeks, and then we allow them to recover, and then combine that with in vivo imaging so that we can monitor the loss and regeneration of oligodendrocytes. And histologically, we can show here, you can see 6 weeks on cuprizone results in complete loss of oligodendrocytes, and even a 3-week treatment results in almost complete loss of oligodendrocytes. So the 2 cells that are highlighted by the blue arrowheads here are the only 2 cells that survive within this field of view. So we typically use the 3-week cuprizone protocol. And this time-lapse series shows you really what happens in the cortex. So there's a period of sort of like decline and shrinkage of the oligodendrocytes before they disappear, and then you see the appearance of these very bright, almost like star-shaped cells. So these are the initial regenerated oligodendrocytes. And the regenerated oligodendrocytes are very bright because before they condense the myelin, they have a lot of cytoplasm, and so their processes are very, very bright for a while until they form the mature myelin sheets. And so we looked at the loss of oligodendrocytes and compared that to what happens in a normal animal. So the horizontal lines here represent individual oligodendrocytes. You can see that these are very stable over the imaging period, and 2 new cells were generated as indicated by the green bars. And this results in a cumulative increase in the total number of oligodendrocytes. So what happens in the cuprizone case? As you might expect, since the cells are dying, so you can see by the termination of these lines, each one of these cells disappeared from the field of view. And as we then took the animals off cuprizone, new cells started to appear. And the regeneration profile is illustrated by the green bars. Now you can appreciate a couple things from this. So one is that there's very little survival of oligodendrocytes within the cortex in response to cuprizone exposure. And there is a really robust regenerative response. But that regenerative response is not complete. So even after 12 weeks here during the experiment, we don't have complete recovery. And the reason that we don't have complete recovery is kind of illustrated here. So you can look at the number of new cells that are generated. There's an initial burst of generation of oligodendrocytes, but that burst is not sustained. And so it's a transient burst that declines over time. And as a result, one doesn't recover the total number of oligodendrocytes. And if you extrapolate the rate at which oligodendrocytes are generated, essentially these animals will never regenerate all of their oligodendrocytes. So even though we kind of think of mice as being really good at regeneration, and indeed most studies of demyelination that have looked at things like lysoleptin injection and so forth have reported really robust regeneration of oligodendrocytes. Here we can see that in this broad model that actually the regeneration is not that efficient. And furthermore, we analyzed the degree to which oligodendrocytes could be regenerated in different regions of the cortex. So what we did is subdivide the cortex into these 3 different bins that were about 100 microns in depth, and then looked at the degree to which the cells were regenerated. And the take-home message from this is that there's a depth-dependent decline in the regeneration of oligodendrocytes. You can see that here, that actually the upper cortical layers recover almost completely. But as you go deeper into the cortex, you get less and less recovery. And that can be seen dramatically here if we plot the number of oligodendrocytes. In gray is the normal distribution, and in green shows the regenerated distribution after 5 weeks. And if you look at the distribution, sort of like, well, it's almost uniform over the entire cortex, even though you have much greater number of oligodendrocytes that are present normally within deeper layers of cortex. So there's a real lag in the efficiency with which oligodendrocytes can be regenerated in deeper layers of the cortex. And one of the things that we think might be going on is that there's evidence for increased gliosis as you go deeper into the cortex. And that can be illustrated by the increase in GFAP immunoreactivity. And if you look at the ratio of the upper layers to the lower layers relative to the GFAP, you can see that the deeper layers show this persistent evidence of gliosis that's very prolonged in these animals in the deeper layers. And what might be happening here is because there's more oligodendrocytes in the deeper layers, when you induce degeneration, you have more myelin debris that's formed and things, and that may provoke greater inflammation, greater gliosis, and that may impair the ability of the progenitor cells to undergo differentiation. So this is something that we're looking into in more detail now. Now, the other thing that we discovered in doing these experiments was something very unexpected, and that was that that the pattern of myelin that is recreated is quite different from what existed before. Now, here's an example where we've marked the individual cell bodies of the oligodendrocytes over time, and in the control animal, all of these cells are stable, and then you have a couple new cells that are formed and kind of inserted within the cortex. Now, what happens when you have degeneration or regeneration is illustrated here. So you can compare the distribution of the magenta cells with the green cells. And strikingly, they are not overlapping. And that is surprising because you think of a regenerative process as one that is designed to restore function. And the simplest way that you would want to do that, one would think, is to basically place oligodendrocytes in the same location where they were before, so they can myelinate the same axons. And what we find is that oligodendrocytes are generated in new locations, not overlapping where they were originally. And this can be seen in these 3D plots. The control is on the left, the green cells are the new cells, and on the right is the plot where the lost cells, the cells that degenerate, are shown in magenta, and the new cells are shown in green, and you can see there's almost no overlap between them. The black circles are the few oligodendrocytes that survive during this protocol. The vast majority of them degenerate, and then they are regenerated at new locations. And we can see that this is quite statistically significant if we look at the nearest neighbor distance. And I'll just highlight this final bar here. If we look at all cells within the upper 100 microns, where there's essentially of the oligodendrocyte population, you can see that the nearest neighbor distance is greatly increased if we compare the original cells to where the new cells are formed. So if they were right on top of each other, the distance would be very small, but they're being generated in locations, so there's really a dramatic increase in this distance. So one possibility is that oligodendrocytes are accessing the same population of axons by changing their morphology. And so we have done detailed reconstructions of oligodendrocytes, now reconstructing the cell body, their individual cytoplasmic processes, and all of the sheaths that are formed by these cells. So it's a really tedious process, but it had to be done to try to understand what happens to the morphology of the cells. And 2 example cells are shown here, and you can appreciate that, well, the remyelinating cells are really similar looking to the new cells. Like they're not highly polarized or anything. And actually, if we now look at the distribution of the sheets, you can see that these cells are actually not more polarized than they were originally. So oligodendrocytes tend to be a little bit polarized. We can see that in these vectors. There's a little bit of polarization, but that's the same polarization that we've seen in the processes of the regenerating cells. So they're really not different. And they would have to be very polarized if they were going to reach over and get the same axons that were myelinated before. And so we then tried to, since we can only reconstruct a few of these cells, we thought, well, maybe we can get some quantitative estimate of the degree to which they can access the same axons. And we did that by creating spheres that encompass the majority of the processes of the cells. So you can see that it's a bit of like an ellipse for the cells, and so we created ellipses, and then we placed the ellipses on the cell bodies of the oligodendrocytes within the volumes. So here's what happens in control. Here's the territory that's occupied by individual oligodendrocytes. Over time in a control animal, you add cells to that population. So what happens in the case of Cooper's where you degenerate? So all of these magenta cells die, the green cells are regenerated, and then you can look at the degree of overlap in the territories between these cells. We found that there was only, maximally about 60% overlap between the territories. So, I guess that's a lot, but it's actually the 40% of it is now just completely non-overlapping. So that would suggest that there's really new territory that's being accessed by these regenerated cells. And then we looked at individual volumes and looked at the sheets in those volumes. And you can see here that most, in a control animal, most of the sheets are very, very stable, as you would expect. And then there's a few new oligodendrocytes that were generated that produced novel sheets. Now, the Cooper's case is dramatically different from that, and you can tell by the colors here. And maybe the most important ones to highlight are the ones that are lost in magenta. So these are myelin sheets that were there originally that were not regenerated. And then the green ones are actually new sheets that were formed along axons that were previously unmyelinated. So it's quite a dramatic change in the overall pattern of myelination. There's no change in the total number of internodes if we compare control animals to Cooper's animals, but a dramatic change in the overall pattern. And if we plot now the proportion of internodes relative to baseline, you can see that very, very stable in control. Cooper's almost all of them are lost. About half of the myelin sheets are replaced in Cooper's about 40% were not replaced, and then there's about half of the sheets that are novel. That is, they're extending processes that are forming sheets on axons that were previously unmyelinated. So what we conclude from this is that oligodendrocyte regeneration creates a new pattern of myelin in the cortex. Now, can you ever replace the same myelin sheet? So we did some high-resolution imaging to look at individual myelin sheets. So here's an example of two sheets separated by a node neuron A. This degenerates, and then remarkably, over time, you can see that two new myelin sheets were formed along this axon, and that the node is almost in the same location that it was before. So here's an example of where it was at baseline, and then after 5 weeks of recovery. So what about the individual isolated myelin sheets? So here's one sheet. The oligodendrocyte that formed that degenerated, and then a new oligodendrocyte, which was actually located in a different location, extended a process, found that same axon, needle in a haystack, formed a sheet there, and the sheet has almost the same location as the original sheet. So it is possible for oligodendrocytes to form sheets in the exact location, but it's very challenging because they are being formed in new locations, and they seem to maybe not encounter those axons with high reliability. How can they form sheets in the same location? Well, one possibility is that there are landmarks along axons which allow this to occur. So we looked at this histologically, immunolabeling for components of nodes such as beta-4 spectrin and Casper. And you can see in this comparison here, control versus Cooper's that a lot of the Casper is lost in these mice, but a lot of the beta-4 spectrum remains. Indeed, these long stripes here are the initial segments along axons, and then there are these dots which form the node of Ranvier. And so you can appreciate by all this immunolabeling that there's a lot of beta-4 spectrum that remains, and it has a distribution, a localization that was consistent where the nodes and axon initial segments were before. So it seems like portions of the nodes remain for a long period of time after demyelination. And this may provide landmarks that can be then accessed by the newly generated oligodendrocytes. This is a plot showing the nodes, heminodes, and isolated nodes, illustrated by this cartoon here. You can see over time, after Cooperzone, there's a gradual loss in the flank nodes because you're losing Casper, but there is still a large number of heminodes and isolated nodes, indicating that these components of the nodes are persisting. It raises a lot of interesting questions about whether or not there could be a critical period for regeneration of these, because eventually one would imagine that some of these things must be internalized and dissolved, and then if you induce remyelination after that, maybe there's no landmarks anymore, which would help it to restore. So finally, I wanted to tell you a little bit about the distribution of myelin along different neuronal subtypes. And one possibility is that because these myelin patterns are changing, you could actually have altered myelin patterns but preserve function, just basically shifting the myelin to different locations along the axon. Or you could just change the amount of myelin that's on one axon versus another, and if this was happening on different cell types, you could really radically change the circuit processing capabilities in the cortex. So we set out to try to understand this, and it turned out it was quite complicated because we first had to define the patterns of myelination along discrete cell types. And the details don't really matter, but we've used genetic approaches and viral labeling approaches to label a number of discrete cell types in the cortex. And you can see here in these plots that things like parvalbumin interneurons and BM, so these are from the BM region of the thalamus, somatosensory interneurons, different types of cortical neurons. They exhibit very different patterns of myelination. And so this is a plot of the proportion of their axons which are myelinated. PB and BDM bound cortical axons are relatively highly myelinated. And then there's just a diversity of myelin patterns along the other types of neurons. So again, highlighting the fact that different types of neurons get myelinated to a different extent. And so then we wanted to know, are these myelin patterns restored after demyelination? And this turned out to be kind of a tricky problem, but what we used is this in vivo imaging approach to do longitudinal imaging in these animals during this demyelination and regeneration experiment. And we combined the genetic cells in the grand scheme of things, and of course there's a lot of territory, because in gray matter there's a lot of axons that are not myelinated. So there's a lot of potential regions for myelination. And I have an asterisk here to remind myself to say that we don't see much remodeling of the myelin sheets, but of course we haven't studied the thickness of the myelin. And so it's possible, and people have proposed that myelin sheets individually might be able to shrink or expand and to change its resistive capacitive properties, and that could certainly affect it. That's not something that we've studied here. I showed you how changes in sensory experience can stimulate the production of new oligodendrocytes, and this is something that's been seen in a number of different paradigms now, including motor learning, which can induce the production of new oligodendrocytes and myelin, and studies from the Bill Richardson's lab and others suggest that production of new oligodendrocytes is actually important for motor learning. So it does seem like this adaptive form of myelination could be a new form of plasticity, new in the sense that we're just realizing that it exists. I showed you how specific regions of axons can be remyelinated, and it seems that there are components of the nodes of Ranvier and axon initial segments that remain along axons for a long period of time after demyelination, and that can provide kind of guideposts which will ensure that myelin is sort of placed back in the same location. One of the most remarkable things that we discovered with this analysis is that the myelin patterns change after demyelination. New cells are regenerated in new locations, and because oligodendrocytes sort of have a fixed morphology, where they drop in is where they myelinate. They're not extending processes over much longer distances or anything like that after regeneration, so they tend to myelinate what's in their local vicinity. Because they're produced in new locations, they're myelinating, in some cases, new axons. Very, very surprising. But we showed that, if we look at the properties of individual cells, we find that their overall myelination patterns are preserved. So the total pattern now along individual axons can change, but if you look at the myelin content that is acquired by these different types of neurons, it is preserved after regeneration. So it tells us a lot about what regeneration is trying to do or is able to do in the nervous system. It doesn't seem like it's trying to replace oligodendrocytes one-to-one, but rather to produce myelin along discrete types of cells. And I think that this has a lot of implications for, the development of therapeutics to promote remyelination and myelin plasticity, and lots and lots of questions that we have left to answer about the mechanisms that control these processes. So with that, I'll end and just acknowledge the really extraordinary people that I've had the benefit to work with over the past few years on this project. Jen Orphan Murphy really pioneered the regeneration studies that I mentioned. She now has her own lab at University of Pennsylvania. Ethan Hughes did many studies of oligodendrocyte progenitors and assisted Jen in these initial studies. He has his own lab at University of Colorado, Denver. Cody Call has been looking at the subtype-specific remyelination that I mentioned and being assisted by Gian Molina Castro in those studies. Cody's now a postdoc at the Ballin Institute and Kelly Monk's lab. And Tiger Zhu is an amazing computational biologist who's been developing these algorithms, programs which allow us to track and visualize myelin sheath dynamics in vivo. And obviously very grateful to support from a variety of sources in these studies. So with that, I'll end. I would be happy to take any questions. Thanks.
Thank you, Dwight. Great. Okay, before I ask any questions, we'll have questions from the audience. Okay, Eduardo, maybe you can close your presentation so that we can see you.
Okay, yeah. How's that? Good.
Thank you very much.
Eduardo, okay.
Great.
I got really curious about this specific meiosis in the infragranular layers. Do you have any clue about it? And is it somehow related to the to changes in the extracellular matrix? Is this meiosis somehow producing a scar or a scar-like effect?
It's a great idea. We haven't, so I know almost nothing about it. Other than what I mentioned, was just basically looking at gliosis from the standpoint of like GFAP upregulation. If you look at IDA1 and MARC, like microglial cells, you'll see they're sort of hot and angry in that region for a long period of time. So there's a couple questions like what exactly is happening there, which I think you're pointing out would be really interesting to know if there is some sort of change in the environment that maybe even extends beyond the gliosis. And what I mentioned was no matter how long you wait, you will not regenerate these cells. And it's very clear that gliosis does resolve over time and all these kinds of things like transient inflammation, it goes away. So, we've been thinking about these kinds of things about the matrix maybe changing, or perhaps even the OPCs themselves being, if you will, traumatized by exposure to that inflammatory environment. So you can think of it as sort of like a PTSD. They were existing in the environment for a period of time and may be altered by exposure to that mix of cytokines and altered matrix that may then irreversibly harm them and prevent them from reliably undergoing differentiation. So this is something we're exploring now by, isolating the OPCs from control animals and animals that have lived in that environment, to then see, are there changes in, their transcriptome and methylome, all those kinds of things. And then also, I think, because this is really exploratory still, we're starting to do spatial transcriptomics. So basically take the cortex and ask, what is happening in gene expression across this distance.
And it is interesting, this idea of the insult making progenitor, right? Then maybe if you put them in culture, then you can test if they have like a change in potential to make this.
Yeah, absolutely. And we know PCs, one, with age, show less and less ability to undergo differentiation. So that's whether is that even regionally, like cells that are OPCs that are within the white matter, differentiation, to migrate, and things like that. It's already like hints that this OPC biology is somewhat flexible. And so I think it's not a huge reach to say that it expose them to a unique thing for MS, because, when people do histology in MS, what is surprising is how many progenitors are still there. So if the progenitors are there, why aren't they undergoing differentiation? But of course, we're just labeling the progenitors with an antibody, right? And we don't really know what those progenitors are. It could be that those progenitors weren't. So we have very crude ways of studying that now. But I think that this is changing rapidly as people are doing, single nuclear sequencing and stuff from human tissue and from other species like marmosets, where you can have MRI, longitudinal MRI of lesions and things like that. So I think we're going to start accumulating that kind of insight into it.
Thank you, Dwight. There's a question from Clarissa Monteiro. Clarissa,
Thank you for the presentation, and it's very interesting because oligodendrocytes, they are depending on a very robust lipid metabolism to synthesize the myelin.
Right, yes.
So how did you think already on changing the diet to see how it would impact, like a high-fat diet or a pro-inflammatory diet? This plasticity?
It's a great question. We have done, I can only tell you, we've done a little bit of work using some of the pro-differentiating agents that are currently in clinical trials. So you might know that, there has been a series of experiments, like one, in MS, people have targeted the immune system effectively, right? So there are ways of suppressing the immune system, preventing immune cell entry to the brain, things like that, and that has really help these patients. But of course, patients present with demyelination, so eventually you have to get remyelination. And so there's been concerted effort to try to develop compounds which will promote the differentiation of the progenitor cells. And so these are screens that have been done in vitro, and there are clinical trials that are ongoing. And so we've taken a few of those compounds, and many of them, for reasons that are not well understood, seem to converge on the cholesterol biosynthesis pathway. So that appears to be sort of like some clinical node in the differentiation of the cells for that sort of assignment. So we have looked at some of these compounds in vivo, and what we have seen is some enhancement of regeneration of oligodendrocytes, but almost exclusively in those regions where it always occurs. So in the upper regions of the cortex where recovery is almost complete, we can see accelerated regeneration, but as we go into the deeper layers of the cortex where there's this suppression of regeneration, it does, most compounds don't appear to overcome that. And that could be a consequence of the fact that the screens that were used identify these compounds were done in vitro. So they were just a highly permissive environment. You're basically just asking, does this compound induce the cell or accelerate the differentiation of the cell? You're not adding in inflammation or changes in the matrix or anything else which exists in that environment. So I really feel like now, those things are not going, those compounds are not going to be sufficient. I think that we're going to really have to have a multi-tiered strategy to reduce the inflammation or restore the environment that these cells live in, and perhaps even reprogram the progenitors that exist in those environments to get this recovery. So it's early days. I don't have any specific, most of those compounds are not targeting metabolism per se, but they are involved in this cholesterol and biosynthesis pathway. So loosely it is metabolic in that sense. So yeah, that clearly is a key regulatory pathway in these cells.
Can I add something?
Sure.
Yeah.
So, and did it check for microglia in this? Because you showed GLKP for phospholipids, but also microglia plays an important role in cleaning the environment.
So they, again, we've only looked at this in a very cursory way. So if you do immunolabeling for IPA1, things like that, you see that the microglial cells are highly activated. They're activated in morphology. They look phagocytic. They have hypertrophy, things like that. So in concert with what you would expect for a general gliosis, it's happening there. And we know that microglia are really important for eating the myelin that's produced. So what we haven't done, for example, single cell sequencing of the cells to see how they're all altered, or to track their phenotype over time during recovery. In some ways, I think the most interesting time point is not that really active phase when there's a lot of regeneration, but it's many, many weeks out where the cells still are not recovering and repairing, and yet there really isn't much inflammation that's visible there. So there's probably a lot of hidden changes in either the environment or the cells or both that needs to be uncovered. But yeah, really interesting direction to go in.
Thank you. Here's a question from Anastasia. Anastasia, are you there? There she is.
Good afternoon, everybody, or good morning in the US. I had a quick question for Dwight, great talk, and it's more of a curiosity, I guess, for those of us working in the field, which is, I think it's pretty well accepted that OLs are very stable. They don't really get replaced under only static conditions. So why do we have so many, or at least why does the mouse have so many? Could it be a matter of strength in numbers to maintain homeostasis? Do you think it's more of a competition model than what we see in epithelial tissues? What's your opinion?
So, are you sure I'll say?
So OPC, but then still in the mouse you still have a pretty robust number of I guess the intermediate, right? We grow myelin maybe, but now we know that they also in white matter. From the standpoint of the cellular statement, what's your opinion?
Right. So yeah, I think the evidence is very strong that oligodendrocytes, once generated, are not crowded. So then the question is, okay, so there is new generation of oligodendrocytes. It's by that, in a year and a half, lots of new oligodendrocytes. The most rapid period of oligodendrogenesis is in the first couple months after birth, and then it slowly declines. So why do we have this huge number of OPCs that persist in CNS? I think this is a really, really interesting question, and my sense is that these cells have functions other than serving as regenerative functions. So they share a lot of features with microglia. If form is compatible with function, and you were to look at microglia and OPC side by side, you would see that they share many, many, many things. So one, they have approximately the same distribution, same density, they tile with each other. They are also the only two really dynamic cells in the CNS. So they move around, they extend their processes, they produce, they're constantly searching. We know that OPCs now can bulk material. They can present antigen on their surface, the way that microglia do. So if I had to guess, I think that the OPCs are actually, just because we call them something, it does not fully encapsulate all of their functions. I think that they might be part of the chemicals that weren't even concerned with microglia. And, they are so well positioned to do that. I mean, they are just everywhere in the CNS. And so as a surveillance unit, they're ideally positioned for that. And then they have this dynamics. So you're absolutely right. The degree to which neural dendrocytes are generated in particularly old or adult animals doesn't make sense from the standpoint of why you need to have so many of these cells around. Well, it's not clear from that alone. And in fact, there are OPCs, the regions of the CNS, where there's no myelin at all. So, in the cerebellar molecular layer, there's no myelinated fibers to find OPCs. So that alone kind of tells you that there might be some other function, which I think is extremely interesting and probably has an important bearing on myelin regeneration. Because if the cells that we're looking at are really, in these lesions or around these lesions are different, if they're really now being pushed towards a different function in the nervous system, maybe they're not really progenitors anymore at all. So I think that that's where we really need to explore and be open-minded about the other functions that these cells might have in the nervous system. And you have to be careful about giving names to things. All the oligodendrocyte precursor cells, but of course that doesn't mean that they couldn't also do other things. And it's very clear that they do in the case of injury, for example. You may know that if you induce a focal lesion in the CNS, these OPCs will become polarized and they will migrate to the site of injury and they will contribute to the formation of real scars. And that behavior has absolutely nothing to do with making oligodendrocytes. In fact, the moment that they become polarized and start to migrate toward the injury site, they are replaced by the surrounding cells. Because that OPCs to the homeostasis. So the fact that they're not seeing the cell anymore shows you that they can also transform into these cells that are presumably important for recovery. So quite interesting cells that probably have multiple functions in the CNS.
Do I have time for just one quick question, Sylvia? Otherwise, fine. Okay. Yeah? I'm sure you're familiar with this is more on the remyelination paradigm, with the studies from the Cellular, the transcriptomic study, right? So there's one paper, and I've discussed this with them, that they hint at the idea that there may be a contribution of the old and mature oligodendrocytes to remyelination, right? It's a very small population, and for me it's very difficult because a mature well, it is terminally differentiated. So unlike the PNS, they don't differentiate. So in vivo, you see no evidence of that in the gray matter.
This is a great question. So I think I can only talk about the model that we studied, right? So we used the superchromosome model, and yes, it's the degeneration organism is extensive, but there are cells that survive. Okay, a few percentage of cells that survive, and basically what we see is that over time they tend to lose sheaths. We have not ever seen one of these cells extend a new cytoplasmic process and then form a new sheath. The only thing that we've seen is that if the cytoplasmic process is retained, that one of these can actually, if it retracts, it can maybe extend along that same trajectory of where the internode was. But that is extremely rare. And quantitatively, almost all the cells retract. So basically, we don't see any extensive evidence for cells that survive this injury. Remember, we're many, many weeks out. The animals are taken off zone, even in these regions where there's really high degree of recovery, there's no evidence of inflammation anymore. We don't ever see these cells extending new cytoplasmic processes and forming new sheaths. So, this is the model that we set. So I can only say that in this model, we do not see that behavior, which, suggests to me that like you say, that they're sort of terminally differentiated. They don't show a lot of that form of plasticity, which I think is also consistent with, the behavior of the cells in the control phase. I mean, they're not, changing their sheaths all the time and regrowing them and doing that kind of thing, which would suggest that that's an intrinsic behavior of the cell. They just don't do that normally. So maybe it's not at all surprising that they don't do that, after they've been injured. They also don't do that. They don't seem to recover that way. Now, there are cells that remain, so you can imagine how increasing survival of these cells, maybe reverting them to a little bit more immature phenotype, could promote that kind of behavior. So I think from the therapeutic standpoint, still quite a possibility. And of course, we have not studied this in human tissue, human MS, so we have no idea if human oligodendrocytes, first of all, in that MS environment, could behave in a different way. But I think it is obviously a very sort of controversial topic here, and I kind of side on I'm on the side of progenitors as being a major, source of regeneration. I think, part of the problems is that they're, with the carbon labeling studies from Streisand's lab, there are a lot of assumptions about that, and we've shown in vivo that it basically involves, DNA replication and cell division as a means to look at the regeneration of the cells. But we've shown in vivo that oligodendrocytes can be generated, in fact, are almost always generated by direct differentiation of the progenitor cells. So without cell division. So if they don't divide and it differentiates into an oligo, by using the carbon labeling, you would not see that you would not identify that as a newly generated oligo. So I think that's a huge limitation of the human studies using that approach. Just, if they directly differentiate, which they do in the mouse, then you can't use that approach to really replicate the cells in that way. So a lot more to be done. It's really interesting.
Yeah, very interesting. Thank you very much. Have a good week.
You too.
Dwight, let me ask you. So you think that MG2 progenitors in the cortex are the main source of oligodendrocytes, but it has been shown that subventricular zone progenitors are able to produce oligodendrocytes, and Arturo Alvarez-Buila has shown some studies showing that if you lesion white matter, so here we're not talking about gray matter, but white matter, that these cells can replace and myelinate white matter axons. So what role do you think this population could have?
This is a great point. Thank you for bringing that up. Yes, so work from Toby Merson and Jim Salzer have shown that these V1 positive progenitors around the ventricles can give rise to new OPCs, basically, and adult animals. And so this seems to be, I wouldn't say it's exclusive because they have shown that this also occurs in the chromosome model, but clearly if you induce demyelination, as you pointed out, the white matter, right above where this, this pool of progenitors is located, that you really get a strong response of these cells. And these cells, they will undergo differentiation in the sense that they become OPCs, they migrate in, and they will myelinate even better than the existing cells that are there, it seems. So it's a really exciting idea that, one, whether that pool could be recruited. I'm a little bit skeptical about whether that occurs in human. This kind of goes back to the same debates about, whether there are, granule cells that are generated in the hippocampus, and the extent to which these progenitors actually exist is still being debated. It definitely declines with age, right? So in adults, it's not meant that, of course, it could be enhanced following injury. But clearly in mouse, if you do a lesion there, you can recruit this population. And it's exciting from the standpoint of, either developing new OPCs or perhaps the idea that you could rejuvenate the existing population of cells by driving them to a little bit younger, less mature phenotype. So that's an exciting idea. I think it's hard for me to imagine that that this would be a substantial means of regeneration in the human nervous system, just given the distances that we're talking about. If you have a lesion, it's literally over here relative to the ventricles. How would those cells sense that there was a lesion? How would they actually migrate over such long distances to get there? Is there even a pool of those progenitors that exists? I think there's just a lot. I think it's a very intriguing idea, and the evidence in mouse is absolutely solid that this occurs. Genetic matrix, and you can show that this exists and things like that. But I would say, I have my own bias that I would love to see this with longitudinal imaging, where we can track those progenitors and watch their behavior relative to the existing OPCs that are there. Now, I showed you that even in the deeper layers of cortex, where you are closer to where these progenitors are found, that there's not that, you're not getting that much recovery. So clearly those cells, even though they may be rejuvenated and have some greater ability to undergo differentiation, they're also not able to overcome whatever constraints exist there. So,
But did you look at white matter or only gray matter?
There's not as much demyelination that occurs in this model there, but we haven't. And yeah, so I think this is something that we've been trying to do for a while now. I think so we can only access the upper layers of the cortex with this new photon imaging. So there are real technical limitations, but there are microscopes, 3-photon microscopes that have been developed, which use even longer wavelengths of light to induce excitation, and the longer wavelengths of light are less sensitive to scattering, and so they can penetrate deeper into the brain. And so that kind of imaging modality may allow us to get down into the white matter to study the dynamics of the cells there. So I think this is the next frontier of this kind of study, definitely to go into different brain regions and to go deeper into the white matter. Yeah, we really don't know that much about the dynamics of the cells of white matter. But
I was wondering that because also a lot of imaging in humans has shown that the white matter has been particularly compromised in psychiatric diseases and several psychiatric diseases.
Yep.
I don't know anything about the gray matter.
Well, this is the thing too, is all of these studies are, almost all I would say, are based on kind of MRI or diffusion tensor imaging, things like that, which is not actually imaging myelin. This is a really important thing to remember, is they're measuring water diffusion, right? It is not something, it's correlated with the orientation of myelin sheaths and the attachment of myelin, but it is not myelin itself. So there are a lot of questions about what do those things mean? Are they?
There's a lot of post-mortem.
Yeah. I think it's harder because it's not longitudinal when we do post-mortem, so you have to compare between people, kind of, I guess. So then it's just there's a lot that I think would need to be done to understand those kinds of dynamics. I think it's really exciting, and with this technology and ways of labeling and maybe pushing the 3-photon imaging, I think we'll be in a position to do things like look at the late development of myelin in the frontal regions of the brain that coincide with maturity, to look at how diseases affect myelination patterns along discrete types of neurons now even. We're just taking broad strokes here and starting to look at myelin patterns along discrete types of neurons. But you can see how it gets really complicated, but really I don't see, maybe the important take-home message here is I don't see a technical limitation really on the horizon or that would prevent us to answer these questions. So I think we will be in a position to do these kinds of studies and gain really that kind of high-resolution insight into what is happening to oligodendrocytes and to the really detailed patterns of myelin that are produced along these different types of neurons. So yeah, it's really an exciting time.
Thank you so much. I think we have abused you.
Not at all.
Sufficiently. We have half an hour of answering questions. I think we're done now. Thank you so much, Dwight.
It was my pleasure.
Thanks.
I appreciate everybody tuning in and with your great questions and insight. Appreciate it. And I hope we get a chance to celebrate afterwards sometime soon. I look forward to it.
Hopefully we will.
Yes.
Thank you.
Thanks, Steve, everyone, and look forward to that opportunity.
Bye-bye.
Okay, bye.
Bye.
fui até pra emergência.
Sério? Foi pra emergência?
Ai, mas porque eu adoro também um medicamento na veia pra acabar logo, entendeu?
Entendi.
Que é do lado da minha casa.